Picture yourself now in a place of perfect comfort. Your body sinks deeper into soft blankets as the weight of the day dissolves with each breath. The room around you has fallen into a gentle darkness, punctuated only by the softest ambient light. Outside, perhaps rain taps rhythmically against windows, or silence wraps around you like a protective cocoon. Your muscles release their tension, starting from your forehead and traveling down through your shoulders, your chest, your limbs. Your breathing slows into a natural, effortless rhythm. Your mind begins to drift, thoughts becoming less sharp, more dreamlike, floating away like leaves on a slow-moving stream. You're standing at that threshold between waking and sleeping, where consciousness gently surrenders to something deeper, something profoundly restorative. And as you cross that threshold, something remarkable begins to happen inside your skull. While you rest in this state of peaceful surrender, your brain embarks on one of the most critical operations it will perform all day. This isn't merely rest in the passive sense we once imagined. Sleep represents an active, purposeful state during which your brain undergoes a comprehensive cleaning process that cannot occur while you're awake. For decades, scientists puzzled over why sleep exists at all. From an evolutionary perspective, sleep seems disadvantageous. During those hours of unconsciousness, our ancestors were vulnerable to predators, unable to find food, incapable of defending territory or nurturing relationships. Sleep demanded that we surrender roughly 1/3 of our existence to a state that appeared, on the surface, to serve no purpose beyond leaving us refreshed. Yet sleep persists across virtually every species that's been studied, from fruit flies to elephants, from microscopic nematodes to massive blue whales. This universality suggested something profound, something essential that made the vulnerability worth accepting. The answer began to emerge in 2012 when Dr. Maiken Nedergaard and her team at the University of Rochester made a discovery that would fundamentally change our understanding of sleep's purpose. Working with mice, they identified a previously unknown system operating within the brain, a waste clearance mechanism that functions primarily during sleep. They called it the glymphatic system, a name that combines the role of glial cells with the lymphatic-like functions this system performs. The glymphatic system works somewhat like the lymphatic system that clears waste from the rest of your body, but with unique architectural features adapted to the brain's specific needs and constraints. Your brain lacks a traditional lymphatic system. While your body uses lymph vessels to drain cellular waste and transport immune cells, your brain exists behind the blood-brain barrier, an elaborate security system that carefully controls what enters and exits neural tissue. This barrier protects your brain from pathogens and toxins in the bloodstream, but it also means your brain needed to evolve alternative methods for waste removal. The glymphatic system represents that alternative, an elegant solution to a challenging problem. This system relies on cerebrospinal fluid, the clear liquid that cushions your brain and spinal cord within their bony protective cases. During your waking hours, this cerebrospinal fluid remains relatively static. But when you sleep, something extraordinary happens. The discovery process itself revealed much about how this system operates. Nedergaard's team injected fluorescent tracers into the cerebrospinal fluid of mice and watched as these glowing molecules moved through the brain. What they observed was nothing short of revolutionary. The cerebrospinal fluid flows into the brain along pathways surrounding arteries, moving through spaces between blood vessels and the surrounding brain tissue. These spaces, called peривascular spaces, act as highways for cerebrospinal fluid to penetrate deep into brain tissue. Once inside, the cerebrospinal fluid mixes with interstitial fluid, the liquid that surrounds individual brain cells, picking up metabolic waste products that have accumulated during waking hours. The fluid then carries this waste out of the brain along pathways surrounding veins, eventually draining into the lymphatic vessels in the neck. The glymphatic system depends heavily on specialized cells called astrocytes. These star-shaped glial cells have end feet that wrap around blood vessels throughout the brain, creating and maintaining the peривascular spaces through which cerebrospinal fluid flows. On their surfaces, astrocytes express high levels of a protein called aquaporin 4 or AQP4, which functions as a water channel. These AQP4 channels facilitate the rapid exchange of fluid between peривascular spaces and the interstitial space around neurons. When researchers genetically modified mice to lack AQP4 channels, glymphatic clearance dropped significantly, demonstrating the critical role these channels play in the brain's cleaning process. The polarization of AQP4 channels, meaning their strategic positioning on specific parts of astrocyte surfaces, appears particularly important for efficient waste clearance. The architecture of this system reveals an intricate network of pathways that extends throughout the entire brain. Cerebrospinal fluid doesn't simply slosh randomly through brain tissue. Instead, it follows organized routes determined by the brain's vascular anatomy and the spatial arrangement of cells. Imagine arterial highways bringing fresh cerebrospinal fluid deep into brain tissue. This fluid percolating through the spaces between cells like water seeping through soil, collecting waste products as it goes, then converging onto venous pathways that carry the contaminated fluid out toward drainage sites. The entire process operates on principles of bulk flow, moving large volumes of fluid through tissue rather than relying solely on diffusion, which would be far too slow to clear waste effectively from an organ as metabolically active as the brain. Research has shown that the volume of extracellular space in the brain, the gaps between cells where interstitial fluid resides, changes dramatically between waking and sleeping states. During sleep, studies in mice revealed that the interstitial space expands by approximately 60%, increasing from about 14% of total brain volume during wakefulness to nearly 23% during sleep. This expansion has profound implications for waste clearance. With more space between cells, cerebrospinal fluid can flow more freely, mixing more effectively with interstitial fluid and carrying away more waste. Think of it like the difference between trying to rinse dirt from tightly packed fabric versus loosely woven mesh. The expansion of interstitial space during sleep creates conditions that favor convective flow, the bulk movement of fluid that efficiently sweeps away accumulated debris. But what triggers this expansion? What signal tells brain cells to shrink slightly, opening up the spaces between them? The answer appears to involve norepinephrine, a neurotransmitter associated with arousal and alertness. During waking hours, neurons release norepinephrine throughout the brain, maintaining the alert, responsive state necessary for interacting with the environment. Norepinephrine keeps interstitial spaces relatively constricted, which may optimize conditions for neural signaling but hampers waste clearance. During sleep, norepinephrine levels drop dramatically. This reduction allows cells to relax their volume, opening up pathways for cerebrospinal fluid to flow. The discovery of norepinephrine's role suggests it may act as a master regulator, coordinating the transition between two functional states of the brain: a waking state optimized for information processing and a sleeping state optimized for maintenance and waste removal. The glymphatic system's activity follows a clear circadian pattern, with studies showing that clearance efficiency can be up to 10 times greater during sleep compared to waking hours. This isn't a gradual change, but rather a dramatic switch, an indication that sleep represents a fundamentally different operational mode for the brain. Interestingly, anesthesia appears to activate the glymphatic system similarly to natural sleep, though not all anesthetics produce this effect equally. Certain anesthetics that more closely mimic the neurochemical environment of natural sleep, particularly those that reduce norepinephrine signaling, promote better glymphatic clearance. This observation has important implications for surgical patients and for our understanding of why anesthesia, despite inducing a sleep-like state, doesn't fully substitute for natural sleep's restorative functions. The discovery of the glymphatic system has sparked intense research interest and some scientific debate. Not all researchers initially accepted the findings, and questions arose about whether the methods used to visualize cerebrospinal fluid flow might have artificially altered the very processes being studied. Some investigators reported difficulty replicating certain aspects of the original findings. However, as more research groups have examined the glymphatic system using various methodologies, including non-invasive imaging techniques in humans, a consensus has emerged that the brain indeed possesses organized pathways for waste clearance that become particularly active during sleep. The controversy has been valuable, spurring more rigorous investigations and leading to more nuanced understandings of how, when, and under what conditions glymphatic clearance operates most effectively. In humans, confirming the existence and function of the glymphatic system presented unique challenges. We can't easily inject fluorescent tracers into human brains to watch them flow as researchers did in mice. Instead, scientists have employed advanced magnetic resonance imaging techniques, tracking naturally occurring contrast or using special MRI sequences that can visualize cerebrospinal fluid movement without injecting anything into the brain or spinal cord. These studies have revealed patterns consistent with glymphatic circulation in human brains, showing that cerebrospinal fluid does indeed flow along perivascular spaces and that this flow changes with sleep state and body position. While we still have much to learn about the human glymphatic system, the evidence strongly supports that the basic architecture and function discovered in animal models apply to human brains as well. The implications of this discovery extend far beyond basic neuroscience. If sleep serves a fundamental waste clearance function, then chronic sleep deprivation doesn't just make us tired. It allows toxic substances to accumulate in our brains. This realization has prompted researchers to investigate whether impaired glymphatic function might contribute to neurodegenerative diseases, particularly those characterized by the accumulation of misfolded proteins. The architecture of sleep, we're learning, isn't designed merely for rest. It's designed for renovation, for the essential housekeeping that keeps our most complex organ functioning optimally across a lifetime. As darkness falls and you drift into sleep, an elaborate maintenance crew begins its nightly rounds through the 3 pounds of tissue inside your skull. This crew doesn't clock in from outside. Rather, it represents a coordinated shift in how your brain operates, transforming from an organ focused on processing information to one focused on self-maintenance. The glymphatic system's night shift involves intricate choreography among billions of cells, countless blood vessels, and vast volumes of fluid, all working in concert to accomplish what cannot be done while you're awake. The process begins as your brain transitions through the stages of falling asleep. During this transition, neural activity patterns change dramatically. The rapid, desynchronized firing of neurons during wakefulness gives way to slower, more synchronized oscillations. Your brain waves, measurable through electroencephalography, shift from the fast beta waves of alert wakefulness to the slower alpha waves of relaxed drowsiness, then to the theta waves of light sleep. As you sink deeper, moving into what researchers call slow-wave sleep or deep sleep, your EEG shows large, slow delta waves rolling through your brain like ocean swells. These synchronized oscillations reflect millions of neurons alternating between periods of activity and silence in unison, a pattern that never occurs during wakefulness. These slow waves appear to play a direct role in glymphatic clearance. Research using advanced imaging techniques has revealed that waves of cerebrospinal fluid flow into the brain in coordination with these neural oscillations. During the silent phases of slow waves, when large populations of neurons temporarily cease firing, blood flow to those brain regions decreases slightly. This decrease in blood volume creates a subtle pressure difference that draws cerebrospinal fluid more forcefully into the brain along perivascular spaces. The rhythmic pulsing of cerebrospinal fluid, synchronized with slow-wave activity, may enhance the efficiency of waste clearance, mixing fluids more thoroughly and driving waste products more effectively toward drainage pathways. The role of arterial pulsations adds another layer of complexity to this process. Your heartbeat creates pressure waves that travel through arteries, causing them to expand and contract rhythmically. In most organs, these pulsations dissipate before reaching the smallest vessels, but the brain's unique anatomy, enclosed within the rigid skull, allows arterial pulsations to propagate throughout the entire vascular tree. These pulsations create a pumping action that helps drive cerebrospinal fluid along perivascular spaces. Studies have shown that dampening arterial pulsations through experimental manipulation reduces glymphatic influx, demonstrating that the mechanical forces generated by your heartbeat contribute significantly to the brain's cleaning process. In essence, every heartbeat serves not just to deliver oxygen and nutrients, but also to pump the brain's waste disposal system. Respiration also influences cerebrospinal fluid flow. The act of breathing creates pressure changes in the chest that affect venous return from the brain. These pressure fluctuations, transmitted to the skull via blood vessels and cerebrospinal fluid spaces, generate slow oscillations in intracranial pressure that can drive fluid movement. Recent research using ultra-fast MRI sequences has shown that forced inspiration, breathing in deeply, particularly enhances cerebrospinal fluid flow in both the ventricles and the subarachnoid space surrounding the brain. This finding suggests that breathing patterns during sleep, particularly the deep, regular breathing characteristic of slow-wave sleep, may optimize glymphatic clearance. The interplay between cardiac pulsations, respiratory oscillations, and neural slow waves creates a complex but coordinated symphony of forces that maximize fluid exchange during sleep. The aquaporin, Four channels that stud astrocyte surfaces deserve closer examination for their critical role in this process. These proteins form pores through cell membranes, allowing water molecules to pass through at rates far exceeding what would occur through the lipid membrane alone. A single AQP4 channel can conduct billions of water molecules per second. Astrocytes express these channels densely on their end feet, the portions of the cell that contact blood vessels, creating a highly permeable interface between perivascular spaces and the bulk of brain tissue. This strategic positioning means that as cerebrospinal fluid flows along perivascular spaces, water and small dissolved molecules can rapidly equilibrate with interstitial fluid throughout the surrounding tissue. The polarization of AQP4 channels appears to decline with age, a finding with troubling implications. As we grow older, AQP4 channels become less precisely localized to astrocyte end feet, spreading more diffusely across cell surfaces. This depolarization correlates with reduced glymphatic function, suggesting that aging gradually impairs the brain's ability to clear waste effectively. The mechanisms driving this age-related change remain under investigation, but the consequences seem clear. Older brains accumulate more metabolic debris, potentially contributing to cognitive decline and increased vulnerability to neurodegenerative disease. Understanding how to maintain or restore proper AQP4 polarization might offer strategies for preserving brain health across the lifespan. The journey of cerebrospinal fluid through the brain follows predictable pathways, but with variations depending on brain region. Generally, fluid flows preferentially along arteries, penetrating deep into tissue, then exits along veins. However, the precise routes, the speed of flow, and the efficiency of waste removal vary across different brain areas. Some regions show more robust glymphatic clearance than others, possibly reflecting differences in vascular density, cellular organization, or metabolic demand. The hippocampus, a structure critical for memory formation and one of the brain regions earliest affected by Alzheimer's disease, shows particularly active glymphatic exchange. This observation raises intriguing questions about whether variations in glymphatic efficiency across brain regions might influence regional vulnerability to disease. The exit routes for waste-laden cerebrospinal fluid have also received increasing attention. After picking up metabolic debris from brain tissue, the fluid must leave the skull and enter the body's general waste disposal systems. Research has identified several exit pathways. Much of the fluid drains along larger veins exiting the brain, eventually emptying into cervical lymph nodes in the neck. Additional drainage occurs along cranial nerves, particularly the olfactory nerves that pass through the cribriform plate at the skull base, and along the optic nerves. Some fluid also exits along the walls of cerebral arteries as they pass through the skull. The discovery of lymphatic vessels in the meninges, the membranes surrounding the brain, revealed another component of the waste disposal system. These meningeal lymphatic vessels collect fluid that has traversed brain tissue and transport it to cervical lymph nodes, completing the journey from deep within the brain to the body's general circulation. The coordination required for all these processes to work efficiently seems almost miraculous. Cerebrospinal fluid must be produced at the right rate by the choroid plexus, specialized tissue within the brain's ventricles. This fluid must flow along the right pathways with sufficient force to penetrate tissue and mix with interstitial fluid. Astrocytes must maintain their AQP4 channels properly positioned. Blood vessels must pulsate normally. Neural activity must transition into the slow oscillations of deep sleep. Drainage pathways must remain patent. The body position must allow optimal flow. Any disruption in this chain can impair the system's effectiveness, allowing waste to accumulate where it doesn't belong. Scientists have begun exploring ways to enhance glymphatic function or compensate for its decline. Some research suggests that certain cardiovascular medications, particularly those that block alpha-1 adrenergic receptors and thereby reduce norepinephrine signaling, might improve glymphatic clearance even during wakefulness. Physical exercise appears to benefit glymphatic function, possibly through effects on cardiovascular fitness that enhance arterial pulsatility or through direct effects on neural tissue. Dietary factors may also play a role, with some evidence suggesting that omega-3 fatty acids, intermittent fasting, and limiting alcohol consumption might support better glymphatic clearance. However, much of this research remains preliminary, and the most proven strategy for maintaining glymphatic function is simply ensuring adequate, high-quality sleep. The glymphatic system's night shift, we're learning, isn't a luxury but a necessity. Just as buildings require janitors to remove trash and maintain cleanliness, your brain requires its nightly cleaning to function optimally. The waste products that accumulate during waking hours aren't merely neutral debris. Many are actively toxic if allowed to persist. The proteins, metabolic byproducts, and other molecules that build up during neural activity can interfere with cell function, damage structures, and trigger inflammatory responses. The brain's night shift exists precisely because these waste products pose genuine threats that must be addressed regularly. Missing even a single night of sleep allows measurable increases in brain waste products, demonstrating how quickly trouble can accumulate when the cleaning system isn't running properly. The waste your brain accumulates during waking hours comprises a diverse collection of molecules, some merely unnecessary, others actively dangerous. Understanding what the glymphatic system removes helps clarify why this cleaning process matters so profoundly for brain health and why impaired clearance can have devastating consequences. The molecular garbage that builds up in your brain during consciousness tells a story about the metabolic cost of thinking, the biochemical price of experience. Perhaps the most infamous piece of molecular garbage is beta amyloid. This protein fragment has achieved celebrity status in neuroscience due to its central role in Alzheimer's disease. Beta amyloid is created when enzymes cut up a larger protein called amyloid precursor protein, or AP, which spans the membranes of neurons. Under normal circumstances, AP is processed in ways that generate harmless, soluble fragments. However, alternative processing pathways produce beta amyloid in various lengths, particularly the 42 amino acid version known as amyloid beta 42. This particular variant has an unfortunate tendency to stick to other copies of itself, forming small clusters called oligomers and eventually large deposits called plaques. In healthy brains, beta amyloid is produced continuously throughout the day as a normal byproduct of neural activity. Neurons use and process AP as part of their regular operations, and some beta amyloid production appears unavoidable. The key to maintaining health lies not in preventing beta amyloid production entirely, which may not be possible, but in clearing it efficiently before it can aggregate into toxic forms. Research has shown that beta amyloid levels in the brain's interstitial fluid rise during waking hours and fall during sleep. This pattern suggests that waking activity generates beta amyloid faster than the brain can clear it through the limited clearance mechanisms available during consciousness, while sleep provides an opportunity for the glymphatic system to catch up, washing away the accumulated protein before it can cause problems. When beta amyloid clearance fails to keep pace with production, either because too much is being produced or because clearance mechanisms are impaired, the protein begins to accumulate. Initially, it forms soluble oligomers, small clusters of beta amyloid molecules that remain dissolved in interstitial fluid but exert toxic effects on nearby neurons. These oligomers interfere with synaptic function, the communication between neurons, leading to memory problems and cognitive difficulties. As oligomers accumulate and grow, they eventually precipitate out of solution, forming the insoluble plaques that characterize Alzheimer's disease. These plaques not only directly damage neurons, but also trigger inflammatory responses as the brain's immune cells attempt to clear them, leading to a cascade of destructive processes. The relationship between sleep, glymphatic clearance, and beta amyloid has been demonstrated through multiple lines of research. Studies in mice engineered to produce human beta amyloid showed that sleep deprivation led to increased beta amyloid accumulation in brain tissue. In humans, even a single night of total sleep deprivation caused beta amyloid levels in cerebrospinal fluid to increase by approximately 30%. More remarkably, chronic poor sleep over years predicts higher beta amyloid burden in brain scans among older adults, suggesting that inadequate sleep across the lifespan gradually allows this protein to build up to dangerous levels. The implication is sobering. Every night you don't sleep well, you may be allowing beta amyloid to gain ground, incrementally increasing your risk for Alzheimer's disease decades in the future. Tau protein represents another critical target for glymphatic clearance. Like beta amyloid, tau plays a starring role in Alzheimer's disease, but it also features prominently in numerous other neurodegenerative conditions collectively called tauopathies. Under normal circumstances, tau helps stabilize microtubules, the structural elements that form the internal scaffolding of neurons and provide tracks along which cargo is transported within cells. Tau is particularly abundant in axons, the long projections through which neurons send signals to other cells. Properly functioning tau binds to microtubules and helps maintain their structure, supporting the efficient transport of materials from the neuronal cell body to distant synaptic terminals. However, tau can become abnormal through a process called hyperphosphorylation, in which excessive phosphate groups are attached to the protein. Hyperphosphorylated tau loses its ability to bind microtubules properly and begins to detach from these structures. Once free-floating, tau molecules can interact with each other, forming tangles of paired helical filaments that accumulate inside neurons. These neurofibrillary tangles directly disrupt cellular function and can eventually kill the neuron. Even before tangles form, soluble abnormal tau appears toxic, interfering with synaptic function and cellular metabolism. Research has revealed that tau, like beta amyloid, is subject to sleep-dependent clearance. Studies in both mice and humans showed that tau levels in brain tissue and cerebrospinal fluid follow a circadian pattern, rising during wakefulness and falling during sleep. Sleep deprivation disrupts this pattern, leading to elevated tau levels. More concerning, Chronic sleep disruption in mice engineered to produce human tau led to increased spread of tau pathology through the brain. Tau can propagate from cell to cell, moving through synaptic connections to invade previously healthy neurons. This spreading pattern explains why neurodegenerative diseases progress over time, gradually affecting more brain regions. Impaired glymphatic clearance may facilitate tau's spread by allowing higher concentrations of the protein to persist in interstitial fluid, increasing the likelihood that it will be taken up by neighboring cells. The relationship between sleep, beta amyloid, and tau appears to be bidirectional and self-reinforcing in destructive ways. Poor sleep leads to accumulation of both proteins. Beta amyloid accumulation can disrupt sleep architecture, reducing the amount of deep slow-wave sleep, precisely the sleep stage when glymphatic clearance is most active. This creates a vicious cycle. Poor sleep allows beta amyloid to accumulate. Accumulated beta amyloid further degrades sleep quality, which allows even more beta amyloid to build up. Tau pathology similarly disrupts sleep, particularly reducing slow-wave sleep as tau tangles accumulate in brain regions involved in sleep regulation. Breaking this cycle requires intervention, but by the time symptoms of Alzheimer's disease appear, the cycle may have been reinforcing itself for decades. Each molecule that accumulates in your brain tells a story of metabolic processes, neural activity, and cellular maintenance. Together, they represent the cumulative burden of being awake, of thinking, feeling, and experiencing the world. The glymphatic system's nightly task is to reset this burden, to provide a clean slate for the next day's activities. Beyond these infamous proteins, the brain generates numerous other waste products during normal metabolic activity. Neurons are extraordinarily metabolically active cells, consuming vast amounts of energy in the form of glucose and oxygen to maintain their electrical gradients, synthesize neurotransmitters, and support synaptic transmission. This intense metabolism inevitably produces byproducts. Lactate, a product of glucose metabolism, accumulates during waking hours when neural activity is high. While lactate can be used as an energy source, excessive levels can acidify tissue and interfere with cellular function. The glymphatic system helps clear excess lactate during sleep, resetting conditions for the next day's activities. Reactive oxygen species, unstable molecules produced as byproducts of cellular respiration, represent another category of waste requiring clearance. These highly reactive molecules can damage proteins, lipids, and DNA if not neutralized or removed. The brain is particularly vulnerable to oxidative damage due to its high metabolic rate and high lipid content. While cells possess antioxidant defenses to neutralize reactive oxygen species, some damage inevitably occurs, generating oxidized molecules that must be removed. The glymphatic system helps clear these damaged molecules during sleep, supporting cellular repair processes. Neurotransmitters and their metabolites also require clearance. While much neurotransmitter reuptake occurs rapidly through specialized transporters at synapses, some neurotransmitters and their breakdown products diffuse into interstitial space. Glutamate, the brain's primary excitatory neurotransmitter, can be toxic at high concentrations, causing neurons to fire excessively and potentially triggering cell death. Maintaining low ambient glutamate levels in interstitial fluid requires constant clearance. Other neurotransmitters, including dopamine, serotonin, and norepinephrine, also produce metabolites as they're broken down, and these metabolites must be removed to prevent accumulation. The brain also sheds cellular debris continuously. As neurons and glial cells go about their functions, they periodically discard worn-out organelles, damaged proteins, and other cellular components. Normally, these materials are broken down within cells through processes like autophagy, but some debris ends up in extracellular space. Additionally, when neurons die, either through normal turnover or due to injury or disease, they release their contents into the surrounding tissue. This cellular debris must be cleared to prevent inflammatory responses and to maintain a healthy tissue environment. Inflammatory molecules represent another important category of waste. Whenever the brain experiences stress, infection, or injury, immune cells release cytokines and other inflammatory mediators. While inflammation serves protective purposes in the short term, chronic inflammation damages tissue. The glymphatic system helps clear inflammatory molecules during sleep, contributing to the resolution of inflammation and supporting tissue recovery. This may be one reason why sleep is so important for recovering from illness or injury. The enhanced waste clearance during sleep helps remove the inflammatory molecules that were necessary for fighting infection or initiating repair, but become harmful if they persist. The sheer volume and diversity of waste products that accumulate in the brain during waking hours underscore the challenge the glymphatic system faces each night. This isn't a matter of removing a single type of molecule, but rather of clearing a complex mixture of proteins, metabolites, oxidized molecules, cellular debris, and inflammatory mediators, all while preserving the delicate chemical environment necessary for neural function. The system must accomplish this cleaning without disrupting the synaptic connections that encode our memories and skills, without damaging the neurons that process information, and without disturbing the glial cells that support neural tissue. It's a remarkable feat of biological engineering. Research has shown that the clearance rates of different molecules vary. Smaller molecules tend to be cleared more rapidly than larger ones, as they diffuse more easily through tissue and pass more readily through intercellular spaces. Beta amyloid and tau, being relatively large proteins, clear more slowly than small metabolites like lactate. This size-dependent clearance may partially explain why proteins like beta amyloid and tau are particularly prone to accumulation. Their slow clearance means that even modest impairments in the glymphatic system can tip the balance toward net accumulation. By contrast, small metabolites clear rapidly enough that even somewhat impaired glymphatic function may still prevent their accumulation to toxic levels. The spatial patterns of waste accumulation in the brain also matter. Some regions generate more metabolic waste than others due to higher levels of neural activity. The hippocampus, for instance, is extremely active during learning and memory formation, generating large amounts of waste. If glymphatic clearance is uniform across the brain, then regions generating more waste might be at higher risk for accumulation. Alternatively, if glymphatic clearance is somehow matched to local metabolic demand, then waste might be cleared more efficiently from highly active regions. Understanding these spatial dynamics remains an active area of research, with implications for understanding why certain brain regions are more vulnerable to neurodegenerative disease than others. The molecular garbage your brain cleans each night isn't just an interesting biochemical phenomenon. It represents a fundamental challenge to maintaining brain health across a lifetime. Every day you're awake, your brain generates waste that, if not cleared, would progressively impair function and eventually cause disease. The glymphatic system's ability to clear this waste during sleep is what allows your brain to start each day with a relatively clean slate, ready to learn, remember, and function optimally. When this cleaning fails, either acutely due to sleep deprivation or chronically due to impaired glymphatic function, the consequences manifest first as subtle cognitive impairments and potentially, over decades, as devastating neurodegenerative disease. Sleep isn't a uniform state, but rather a dynamic process cycling through distinct stages, each characterized by unique patterns of brain activity, body physiology, and, it appears, different contributions to glymphatic clearance and waste removal. Understanding when during the night your brain conducts its most intensive cleaning helps illuminate why not just sleep quantity, but also sleep quality matters so profoundly for brain health. The architecture of a normal night's sleep reveals an elegant choreography designed to accomplish multiple essential functions, with waste clearance featuring prominently among them. A typical night of sleep for an adult consists of 4 to 6 complete sleep cycles, each lasting approximately 90 minutes. Within each cycle, your brain progresses through several distinct stages. After falling asleep, you enter stage 1 of non-rapid eye movement sleep, or NREM sleep, a brief transitional period lasting only a few minutes. You then move into stage 2 NREM sleep, which comprises roughly half of total sleep time across the night and is characterized by specific electrical patterns in brain waves called sleep spindles and K-complexes. From stage 2, you descend into stage 3 NREM sleep, also known as slow-wave sleep or deep sleep, characterized by large, slow delta waves rolling through the brain. After spending time in deep sleep, your brain ascends back through stage 2 before entering rapid eye movement sleep, or REM sleep, during which brain activity increases dramatically and most vivid dreaming occurs. After REM sleep, the cycle begins again, with each successive cycle typically containing less deep sleep and more REM sleep. Deep slow-wave sleep appears to be the most crucial stage for glymphatic clearance. Multiple lines of evidence support this conclusion. The expansion of interstitial space, that critical increase in the gaps between brain cells that allows cerebrospinal fluid to flow more freely, is most pronounced during slow-wave sleep. Studies measuring this expansion have found that it correlates specifically with slow-wave sleep rather than other sleep stages. The slow, synchronized oscillations characteristic of this sleep stage create rhythmic pulsations in cerebrospinal fluid flow, with fluid rushing into the brain during the periods when neurons are silent, enhancing the efficiency of waste removal. The dramatic reduction in norepinephrine levels that permits interstitial space expansion reaches its nadir during slow-wave sleep, further optimizing conditions for glymphatic clearance. Research using advanced imaging techniques has directly measured cerebrospinal fluid flow during different sleep stages in humans. One particularly elegant study used functional MRI in combination with electroencephalography to track both sleep stage and fluid movement simultaneously. The results showed that during wakefulness, cerebrospinal fluid exhibited only small amplitude oscillations with a relatively high frequency. However, during sleep, particularly during periods rich in slow-wave activity, large oscillations in cerebrospinal fluid flow occurred at much lower frequencies, roughly every 20 seconds. These large, slow oscillations drove significantly greater inflow of cerebrospinal fluid into brain tissue compared to waking periods. The coupling between neural slow waves and cerebrospinal fluid oscillations was particularly tight during the deepest phases of sleep, suggesting coordinated activity between neural circuits and fluid dynamics. The importance of slow-wave sleep for glymphatic clearance helps explain why this sleep stage seems so essential for cognitive health. Across the lifespan, the amount of slow-wave sleep we obtain naturally declines. Older adults spend far less time in deep sleep than children or young adults, with some elderly individuals obtaining almost no slow-wave sleep at all. This age-related decline coincides with declining glymphatic function, reduced clearance of beta amyloid and tau, and increasing risk for neurodegenerative disease. Whether the reduction in slow-wave sleep causes impaired glymphatic clearance or whether both result from some underlying age-related change in brain structure or function remains debated. However, the correlation is concerning and has prompted investigations into whether interventions that increase slow-wave sleep might improve glymphatic clearance and potentially reduce dementia risk. REM sleep's role in waste clearance appears more complex. While REM sleep clearly serves important functions, particularly for memory consolidation and emotional processing, its contribution to glymphatic clearance seems less robust than that of slow-wave sleep. During REM sleep, brain activity increases dramatically, approaching levels seen during wakefulness. Norepinephrine levels, while not as high as during waking, rise above the very low levels seen during slow-wave sleep. The interstitial space may shrink somewhat compared to slow-wave sleep, as norepinephrine's effects partially reverse the expansion that occurred during deep sleep. These factors would all tend to reduce glymphatic efficiency during REM sleep compared to slow-wave sleep. However, REM sleep likely still contributes to waste clearance in meaningful ways. Even with somewhat elevated norepinephrine and possibly reduced interstitial space expansion, REM sleep maintains conditions far more favorable for glymphatic clearance than waking does. Additionally, REM sleep may support other aspects of brain maintenance that complement waste clearance. Research has shown that REM sleep plays a crucial role in recalibrating neural activity and supporting memory consolidation. Studies indicate that neural population dynamics during REM sleep facilitate the integration of newly learned information with existing knowledge networks. The emotional content of experiences appears particularly dependent on REM sleep for proper consolidation. Some researchers have proposed that different sleep stages serve different but complementary maintenance functions. Slow-wave sleep focuses on bulk waste clearance and global synaptic downscaling, while REM sleep focuses on selective synaptic strengthening and integration of new information with existing memories. Light sleep, particularly stage 2 in REM sleep, comprises a substantial portion of total sleep time and shouldn't be dismissed as unimportant. While it may not feature the dramatic interstitial space expansion of slow-wave sleep or the intense neural activity of REM sleep, stage 2 sleep appears to support glymphatic function through its sleep spindles and K-complexes, distinctive electrical patterns that characterize this stage. Sleep spindles, brief bursts of brain activity, have been linked not only to memory consolidation, but also to maintaining the sleeping state itself, protecting sleep from disruption by external stimuli. K-complexes, large electrical waveforms that appear spontaneously or in response to stimuli, may help regulate cortical excitability. Together, these features of light sleep help maintain conditions conducive to glymphatic clearance while serving additional functions. The progression through sleep stages follows a carefully orchestrated pattern. In the first sleep cycle of the night, you spend relatively little time in REM sleep, but substantial time in deep slow-wave sleep. This early emphasis on slow-wave sleep makes sense if waste clearance is a primary function of this stage. After a full day of waking activity, the brain has accumulated significant metabolic waste that needs clearing. The first few hours of sleep, rich in slow-wave activity, provide an intensive cleaning period when the glymphatic system operates at peak efficiency. As the night progresses and successive sleep cycles unfold, each cycle typically contains less slow-wave sleep and more REM sleep. By the final sleep cycles before morning, you may obtain almost no slow-wave sleep, spending most of the cycle in stage 2 and REM sleep. This changing composition of sleep cycles across the night reflects the different functions being prioritized at different times. Early night sleep, dominated by slow waves, emphasizes physical restoration and waste clearance. Late night sleep, rich in REM, emphasizes memory consolidation and emotional processing. Both are essential. This is why getting only the first half or second half of a night's sleep leaves you feeling different types of impairment. Cutting sleep short by waking too early deprives you primarily of REM sleep, which may leave you feeling emotionally reactive and may impair your ability to consolidate memories effectively. Going to bed very late and sleeping into the day may provide adequate REM sleep, but insufficient slow-wave sleep, potentially impairing waste clearance and physical restoration. The duration of sleep matters profoundly for glymphatic clearance. While the system activates during sleep regardless of duration, clearing the full burden of waste accumulated during waking hours requires adequate time. Studies tracking waste protein levels have shown that even after several hours of sleep, clearance continues, suggesting that a full night's sleep is necessary to restore the brain to optimal cleanliness. Short sleep durations, even if they include some deep sleep, simply don't provide enough time for complete waste removal. This may explain why chronic short sleep, even when it doesn't feel particularly problematic, accumulates health risks over time. Each shortened night allows a bit more waste to remain, and over months and years, this burden grows. The timing of sleep relative to your circadian rhythm also influences glymphatic efficiency. Your body's internal biological clock, synchronized primarily by exposure to light and darkness, orchestrates daily rhythms and hormone release, body temperature, alertness, and numerous other physiological processes. Sleep that occurs at the wrong circadian phase, such as daytime sleep after night shift work, may not provide the same quality of waste clearance as sleep that occurs during your biological night. Some evidence suggests that circadian misalignment impairs glymphatic function, possibly because the coordinated changes in brain state that optimize clearance depend not just on being asleep, but on being asleep at the right time. Shift workers, whose sleep schedules conflict with their circadian rhythms, show elevated markers of brain waste accumulation and face increased risks for neurodegenerative disease, potentially reflecting chronically impaired glymphatic clearance. Sleep continuity represents another critical factor. Fragmented sleep, punctuated by frequent awakenings or arousals that shift you to lighter sleep stages, may significantly impair glymphatic function even if total sleep time seems adequate. Each arousal potentially disrupts the carefully coordinated processes of waste clearance. The interstitial space may contract, norepinephrine levels may spike, and cerebrospinal fluid flow may diminish. If these disruptions occur repeatedly throughout the night, the cumulative effect could be substantial impairment of waste removal despite spending enough total time in bed. This helps explain why conditions like sleep apnea, which fragment sleep through repeated breathing disruptions, carry such significant health risks despite potentially normal total sleep time. The architecture of sleep reveals a system exquisitely designed to balance multiple competing needs. Your brain must process information during wakefulness, which generates waste that must be cleared. It must consolidate memories and process emotions, which require specific sleep stages. It must maintain synaptic plasticity, allowing learning while preventing runaway excitation. It must restore energy stores, repair cellular damage, and regulate immune function. Sleep's stage structure allows all these functions to occur, each during the phase when conditions are optimal. The timing and coordination of these stages across the night reflect millions of years of evolutionary refinement, creating a system that, when allowed to function properly, maintains brain health remarkably well across decades of life. While your brain cleans itself during sleep, it simultaneously engages in equally essential work sculpting your memories and recalibrating your synapses. These processes, far from being separate from waste clearance, appear intimately connected. Understanding how sleep transforms your daily experiences into lasting memories while maintaining synaptic balance reveals the elegant integration of brain maintenance functions. Memory consolidation refers to the process through which initially fragile memories become stable and integrated into long-term storage. When you experience something new during the day, whether learning a fact, acquiring a skill, or experiencing an event, that information initially exists in a labile state, vulnerable to interference and forgetting. The neural patterns activated during learning persist temporarily, but without consolidation, they fade. Sleep provides optimal conditions for transforming these temporary patterns into stable, lasting memories. The process begins with encoding during wakefulness. As you encounter new information, specific populations of neurons fire in particular patterns. These patterns of activity represent the information being processed. Through mechanisms involving changes in synaptic strength, the connections between neurons that fired together become stronger, following the principle that neurons that fire together wire together. This synaptic strengthening creates a physical trace of the experience in your brain. However, this initial trace remains relatively weak and local, typically centered in the hippocampus, a structure crucial for forming new memories. During sleep, particularly during slow-wave sleep, the hippocampus begins replaying these neural patterns. Studies recording from hippocampal neurons in sleeping animals have detected these replay events, showing that the same sequences of neural firing that occurred during waking experiences repeat during sleep, often at accelerated speeds. This replay appears to serve as a teaching signal to the neocortex, the outer layer of the brain where long-term memories eventually reside. Through repeated replay, the hippocampus gradually trains neocortical networks to represent the memory independently, a process called systems consolidation. Over time, as the memory becomes consolidated in neocortical networks, dependence on the hippocampus decreases, and the memory becomes more permanent and integrated with existing knowledge. The coordination between hippocampal replay and neocortical processing happens through specific brain rhythms during slow-wave sleep. When hippocampal neurons replay a memory, they often do so during brief, high-frequency oscillations called ripples. These ripples tend to occur during the up state of slow waves, when neocortical neurons are depolarized and receptive to input. Additionally, sleep spindles, brief bursts of brain activity generated in the thalamus and propagated to the neocortex, often coincide with hippocampal ripples. This precise temporal coordination, sometimes called ripple spindle coupling, creates windows of enhanced communication between the hippocampus and neocortex, facilitating the transfer of information from temporary to permanent storage. The relationship between these memory consolidation processes and glymphatic clearance appears more than coincidental. Both processes require the brain to enter a fundamentally different operational mode from wakefulness. Both depend critically on slow-wave sleep. Both seem to benefit from the expansion of interstitial space and the reduction in norepinephrine levels that characterize sleep. It's possible that the two processes are mechanistically linked, that the same changes in brain state that enable efficient waste clearance also create optimal conditions for memory consolidation. Consider the role of synaptic activity in generating metabolic waste. Active synapses consume energy and produce waste products. During learning, when synapses are being modified and strengthened, metabolic demand increases. The waste generated during intense learning and memory encoding may need to be cleared to prevent it from interfering with the consolidation process. Conversely, the consolidation process itself, with its patterns of neural replay and synaptic modification, generates additional metabolic byproducts requiring clearance. The sleep processes of memory consolidation and waste clearance may thus be inherently intertwined, each supporting the other. REM sleep contributes distinctively to memory consolidation, particularly for emotional memories and procedural skills. Research has shown that emotional experiences receive preferential consolidation during REM sleep, possibly because the neurochemical environment of REM, characterized by high acetylcholine but low norepinephrine and serotonin, creates conditions that favor the processing of emotional content while damping the stress response that might otherwise be triggered by recalling emotional or threatening experiences. This may explain why REM sleep appears to help process traumatic experiences, allowing the emotional charge to dissipate while preserving the memory of what happened. Procedural memories, those involving skills and habits rather than facts and events, also benefit particularly from REM sleep. Learning a new motor skill, whether playing an instrument, typing, or performing a sports technique, involves gradual refinement through practice. During REM sleep, the neural circuits involved in the skill show reactivation patterns, and measures of REM sleep correlate with subsequent improvement in skill performance. The theta rhythms characteristic of REM sleep may facilitate the synaptic plasticity necessary for skill refinement, allowing practice sequences to become more automatic and efficient. Beyond consolidating specific memories, sleep serves a broader function of synaptic homeostasis, maintaining the overall balance of synaptic strength across the brain. During waking hours, learning and experience lead to net synaptic strengthening. Neural circuits involved in whatever you're doing, thinking about, or paying attention to undergo synaptic potentiation, increasing their connectivity and excitability. While this strengthening is necessary for learning, unchecked synaptic potentiation would pose problems. Stronger synapses consume more energy, take up more physical space, and increase neural excitability. If synaptic strengthening continued without any counterbalancing process, circuits would eventually become saturated, unable to learn new information, consuming unsustainable amounts of energy, and potentially prone to excessive excitability or seizures. The synaptic homeostasis hypothesis proposes that sleep serves to downscale synaptic strength globally, reducing the overall burden while preserving relative differences that encode important information. During slow-wave sleep, the slow oscillations that sweep through the neocortex may drive this downscaling process. The rhythmic alternation between upstates and downstates, between periods of neuronal firing and silence, creates conditions that favor synaptic depression, a weakening of connections. However, this downscaling is not uniform. Synapses that were strongly potentiated during waking, those involved in important learning, show relative resistance to downscaling or downscale less than weakly potentiated synapses. The result is a renormalization of synaptic strength. The overall burden decreases, but the signal, the pattern of relative synaptic strengths that encodes memories and learned behaviors, remains intact or even becomes sharper. Evidence for synaptic homeostasis during sleep comes from multiple sources. Electron microscopy studies examining synaptic structures in animals before and after sleep show that the physical size of synapses, which correlates with their strength, decreases during sleep and increases during waking. Electrophysiological measures of synaptic responses show similar patterns, with responses stronger after waking than after sleep. Molecular markers of synaptic potentiation, proteins that accumulate in strengthened synapses, show higher levels after waking than after sleep. Together, these findings support the idea that sleep actively reduces synaptic strength, counterbalancing the strengthening that occurs during waking. This downscaling serves multiple important functions. By reducing overall synaptic strength, it decreases energy demands, allowing the brain to function more efficiently. By reducing the physical size of synapses, it frees up space and resources for future learning. By reducing overall excitability, it may help prevent excessive neural activity and maintain stable circuit dynamics. And by preserving relative differences while reducing absolute strength, it enhances the signal-to-noise ratio of stored information, potentially improving memory clarity and recall. The integration of waste clearance, memory consolidation, and synaptic homeostasis during sleep reveals a picture of brain maintenance far more sophisticated than simple rest. Sleep represents a carefully orchestrated state during which the brain performs multiple essential functions simultaneously. The same slow waves that drive glymphatic clearance also facilitate memory replay and synaptic downscaling. The expansion of interstitial space that enables waste removal may also provide space for synaptic restructuring. The reduction in norepinephrine that allows glymphatic flow may also create the neurochemical conditions necessary for effective memory consolidation. These interconnected processes highlight why sleep cannot be adequately replaced by quiet rest or by pharmacologically induced unconsciousness. True restorative sleep requires the natural progression through sleep stages, the coordinated oscillations of neural activity, the precisely timed changes in neurotransmitter levels, and the activation of glymphatic flow. Disrupting any element of this complex system can impair all the others. Poor waste clearance may interfere with memory consolidation. Inadequate memory consolidation may prevent proper synaptic downscaling. Impaired synaptic homeostasis may compromise subsequent learning and may interfere with sleep quality itself, creating a self-reinforcing cycle of dysfunction. The memories you consolidate during sleep don't simply transfer unchanged from temporary to permanent storage. Rather, sleep actively transforms memories, extracting general principles, integrating new information with existing knowledge, and even facilitating creative insights. The old saying about sleeping on a problem to find a solution reflects a genuine phenomenon. During sleep, particularly during REM sleep, the brain can form novel associations, connect disparate pieces of information, and reorganize knowledge in ways that lead to insights and creative breakthroughs. This transformative aspect of sleep-dependent memory processing reveals that sleep isn't merely preserving the day's experiences, but actively working with them, refining them, finding their meaning and their place within the larger context of your knowledge and experience. The synaptic changes that occur during sleep also influence how you learn the next day. A well-rested brain, with properly calibrated synaptic strength, is prepared to encode new information efficiently. Sleep-deprived brains, with saturated synapses and impaired plasticity mechanisms, struggle to form new memories. Studies have shown that sleep deprivation before learning impairs the brain's ability to activate regions necessary for encoding, particularly in the hippocampus. The neural substrates of learning literally cannot respond normally to new information when you haven't slept. This provides a neurobiological explanation for why pulling all-nighters to study is counterproductive. Without sleep to reset your synapses, your brain cannot effectively learn the material you're trying to cram. When the glymphatic system cannot operate effectively, whether due to inadequate sleep, poor sleep quality, or impaired clearance mechanisms, the consequences ripple through virtually every aspect of brain function. Understanding these consequences reveals why sleep isn't optional, why chronically sacrificing sleep carries real and substantial costs to cognitive performance, emotional regulation, and long-term brain health. The immediate effects of sleep deprivation on cognitive function are both measurable and substantial. After just one night of total sleep deprivation, cognitive performance declines markedly across virtually all domains. Attention becomes harder to maintain, with more frequent lapses and slower reaction times. Working memory, your ability to hold and manipulate information temporarily, shows significant impairment. Decision-making abilities deteriorate, with people making riskier choices and showing impaired judgment. Even a single night without sleep produces cognitive impairments comparable to legal intoxication, with studies showing that 24 hours of wakefulness produces performance deficits equivalent to having a blood alcohol concentration of 0.1%, above the legal limit for driving in most jurisdictions. The brain changes underlying these cognitive impairments are visible in neuroimaging studies. Sleep-deprived brains show altered activity patterns across multiple regions. The prefrontal cortex, responsible for executive functions like planning, decision-making, and impulse control, typically shows reduced activity after sleep loss. This hypoactivity of prefrontal regions helps explain the poor judgment and impulsivity often seen in sleep-deprived individuals. Simultaneously, other brain regions may show increased or erratic activity. The default mode network, a set of brain regions typically active during rest and mind wandering, often fails to deactivate properly during tasks in sleep-deprived individuals, potentially reflecting impaired ability to focus attention on external demands. Emotionally, sleep deprivation produces profound effects. The amygdala, a brain structure central to emotional processing, shows heightened reactivity in sleep-deprived individuals, responding more strongly to negative stimuli. Simultaneously, connections between the amygdala and prefrontal cortex, which normally help regulate emotional responses, weaken. This combination creates a state of emotional hyperreactivity with impaired emotional control. Sleep-deprived people often report feeling more irritable, experiencing mood swings, and finding it harder to cope with stress. They're more likely to experience conflicts in relationships and to make emotionally driven decisions. The emotional dysregulation caused by sleep deprivation can be profound enough to mimic or exacerbate psychiatric conditions like anxiety and depression. The relationship between sleep and mental health is bidirectional and complex. Poor sleep can trigger or worsen depression and anxiety, while these conditions often disrupt sleep, creating self-reinforcing cycles. Chronic sleep deprivation is a significant risk factor for developing mood disorders, with some research suggesting that persistent insomnia may predict subsequent onset of major depression. The mechanisms likely involve both the direct neurobiological effects of sleep loss on mood-regulating brain circuits and the indirect effects of impaired cognitive function and emotional regulation on one's ability to cope with life stressors. The memory impairments caused by sleep deprivation extend beyond simple forgetfulness. Sleep-deprived individuals not only struggle to form new memories, but may also be prone to forming false memories, remembering events that didn't occur or distorting details of actual events. The mechanisms underlying false memory formation likely relate to impaired encoding precision and impaired ability to distinguish similar experiences. When the hippocampus cannot precisely encode event details due to sleep loss, memories become fuzzy and susceptible to confusion with other similar experiences or with imagined events. Chronic partial sleep restriction, getting insufficient sleep night after night, produces cumulative cognitive deficits that worsen over time. While people often believe they adapt to short sleep and function normally, objective testing reveals progressive impairment. After just a week of sleeping 6 hours per night, cognitive performance decreases to levels equivalent to 2 nights of total sleep deprivation, even though individuals typically underestimate their impairment. This disconnect between perceived and actual performance is particularly concerning because it means people operating in impaired states often don't recognize their deficits, leading to poor decisions about when it's safe to drive, make important decisions, or perform critical tasks. The long-term consequences of chronic sleep deprivation for brain health are increasingly recognized as severe. Epidemiological studies consistently show associations between chronic short sleep and increased risk for multiple neurodegenerative conditions, particularly Alzheimer's disease. People who regularly sleep fewer than 7 hours per night show higher rates of dementia decades later. While these studies cannot prove causation, experimental evidence linking sleep to waste clearance provides a plausible mechanism. Years of inadequate sleep allow progressive accumulation of toxic proteins like beta amyloid and tau, eventually reaching levels that trigger neurodegeneration. Brain imaging studies support this connection. Research using PET scans to measure beta amyloid burden in living brains has shown that people with chronic sleep problems accumulate more beta amyloid over time. Even in middle-aged adults who are decades away from potential Alzheimer's symptoms, poor sleep quality and short sleep duration correlate with higher brain beta-amyloid levels. These findings suggest that the damage begins accumulating long before symptoms appear, emphasizing the importance of maintaining healthy sleep habits throughout adulthood rather than waiting until cognitive problems emerge. The vicious cycles that develop when sleep problems and brain pathology interact are particularly concerning. As mentioned earlier, beta-amyloid accumulation disrupts sleep, particularly reducing slow-wave sleep. This creates a positive feedback loop. Poor sleep allows beta-amyloid accumulation, which further disrupts sleep, allowing more accumulation. Once established, such cycles may be difficult to break. This may explain why interventions to improve sleep in people already showing early signs of Alzheimer's pathology often show limited benefit. The underlying pathology has already disrupted the sleep systems needed for effective clearance. Sleep apnea, a condition where breathing repeatedly stops during sleep, provides a particularly stark example of how disrupted sleep affects brain health. Sleep apnea fragments sleep through repeated arousals and causes intermittent hypoxia, reduced oxygen levels in the brain. The combination of sleep fragmentation, impaired glymphatic clearance, and direct effects of oxygen deprivation creates substantial brain health risks. People with untreated sleep apnea show accelerated cognitive decline, increased risk for dementia, increased stroke risk, and structural brain changes including reduced gray matter volume in key regions. Treating sleep apnea with interventions like continuous positive airway pressure can improve symptoms and may slow cognitive decline, but whether it can reverse existing damage remains unclear. The effects of sleep deprivation on basic cellular health in the brain add another layer of concern. Sleep-deprived neurons show signs of stress, including reduced expression of genes involved in synaptic function and increased expression of stress response genes. Some research suggests that prolonged sleep deprivation can lead to actual neuronal death in certain brain regions. Studies in animals have found that extended sleep deprivation causes loss of neurons in the brain stem and other areas. While such extreme sleep deprivation is rare in humans, the findings raise concerning questions about whether chronic moderate sleep restriction might cause gradual cumulative damage to neurons. The protein pleiotrophin, identified in recent research as declining with sleep deprivation, appears to play a protective role for neurons. When pleiotrophin levels drop due to sleep loss, neurons become more vulnerable to damage. This finding suggests that sleep isn't merely removing bad things, but is also actively maintaining the expression of protective factors. Sleep deprivation thus represents a double hit. Waste accumulates while protective mechanisms weaken. The impact of poor sleep extends beyond neurons to affect the very cells that enable glymphatic clearance. Astrocytes, the cells whose aquaporin-4 channels facilitate fluid exchange, show altered function after sleep deprivation. Their ability to regulate ion and fluid balance in the brain becomes impaired. The glymphatic system itself appears to function less efficiently after periods of sleep deprivation, creating another vicious cycle. Lack of sleep impairs the system that's supposed to clean the brain during sleep, making subsequent sleep less restorative. Inflammatory changes in the brain represent another consequence of inadequate sleep. Sleep deprivation activates microglia, the brain's immune cells, pushing them toward an inflammatory state. These activated microglia release inflammatory molecules that can damage neurons and impair synaptic function. Chronic inflammation in the brain, sometimes called neuroinflammation, is increasingly recognized as contributing to multiple neurological and psychiatric conditions. Sleep's role in resolving inflammation and maintaining microglia in a healthy state highlights another mechanism through which poor sleep damages brain health. The cardiovascular effects of sleep deprivation also indirectly affect brain health. Chronic short sleep increases risk for hypertension, heart disease, and stroke. These cardiovascular conditions compromise blood flow to the brain, potentially impairing glymphatic function, which depends on arterial pulsations, and directly damaging brain tissue. The relationship between sleep, cardiovascular health, and brain health forms interconnected networks of cause and effect, where problems in one domain cascade into others. Metabolic dysfunction triggered by sleep deprivation further compounds brain health risks. Sleep loss impairs glucose metabolism and insulin sensitivity, increasing risk for type 2 diabetes. The brain depends heavily on glucose for energy, and impaired glucose metabolism can compromise neural function. Additionally, diabetes itself increases risk for dementia, possibly through effects on cerebral blood vessels and through mechanisms involving protein glycation and inflammation. Sleep deprivation's metabolic effects thus create additional pathways through which it can harm long-term brain health. The cognitive reserve hypothesis, which proposes that brains with more robust neural networks can better withstand damage before symptoms appear, takes on new meaning in light of sleep's importance. Building cognitive reserve requires learning, engaging in mentally stimulating activities, maintaining social connections, and keeping the brain healthy through good nutrition and exercise. But all of these reserve-building activities may be undermined if sleep is inadequate. Without sleep to consolidate learning, clear waste, and maintain synaptic health, efforts to build cognitive reserve may be like trying to fill a bucket with holes in it. The cognitive investments you make during the day require sleep to yield their full returns. Children and adolescents may be particularly vulnerable to sleep deprivation's effects on the brain. During these periods of ongoing brain development, sleep plays additional crucial roles in supporting the growth and refinement of neural circuits. Sleep deprivation during development may have consequences that extend beyond the immediate period of lost sleep, potentially altering the trajectory of brain maturation. Studies have shown that adolescents who chronically don't get adequate sleep show poorer academic performance, increased risk for mental health problems, and may show lasting effects on brain structure. The concerning trend toward later sleep times and earlier school start times in many societies creates widespread chronic sleep deprivation in adolescents precisely when their brains are undergoing critical developmental processes. Given the critical importance of glymphatic clearance for brain health, the question naturally arises: what can you do to optimize this process? While research in this area continues to evolve, several factors have been identified as influencing glymphatic efficiency, offering practical strategies for supporting your brain's nightly cleaning cycle. Sleep position represents one of the more surprising findings about glymphatic function. Research in rodents found that sleeping in a lateral position, lying on the side rather than on the back or stomach, enhanced glymphatic clearance compared to other positions. The mechanism likely involves effects on the flow dynamics of cerebrospinal fluid, with the lateral position apparently providing more efficient drainage pathways. In humans, most people naturally shift positions throughout the night, but those who can comfortably sleep on their side may be optimizing their glymphatic clearance. Interestingly, Surveys of sleep positions across cultures find that side sleeping is most common, suggesting that humans may have naturally gravitated toward positions that support this important biological function. Sleep duration stands as perhaps the most straightforward factor affecting glymphatic clearance. The system needs adequate time to clear the waste accumulated during waking hours. While individual needs vary somewhat, most adults require 7 to 9 hours of sleep per night for optimal health. Consistently sleeping less than this allows incomplete waste clearance, with each night's deficit potentially accumulating over time. Unfortunately, modern lifestyles often normalize insufficient sleep, with many people sleeping well below recommended amounts. Prioritizing sleep duration, treating it as non-negotiable rather than as something to sacrifice when other demands arise, represents a fundamental investment in brain health. Sleep quality matters as much as quantity. Fragmented sleep, interrupted by frequent awakenings or respiratory disruptions, impairs glymphatic function even if total time in bed seems adequate. Factors that fragment sleep include sleep disorders like sleep apnea and restless leg syndrome, environmental disruptions like noise or light, excessive alcohol consumption, certain medications, and anxiety or stress. Addressing these factors can substantially improve sleep quality. For sleep disorders, professional evaluation and treatment are essential. For environmental factors, creating an optimal sleep environment can help. A quiet, dark, cool bedroom promotes uninterrupted sleep. For stress and anxiety, developing relaxation techniques and addressing underlying causes can improve sleep continuity. The timing of sleep relative to your circadian rhythm influences sleep quality and glymphatic efficiency. Your biological clock, synchronized primarily by light exposure, determines when your body is primed for sleep. Sleeping at consistent times that align with your natural circadian tendencies supports better sleep architecture and more efficient restorative processes. Irregular sleep schedules, frequent time zone changes, and shift work that requires sleeping during biological day all disrupt circadian alignment and may impair glymphatic function. While not everyone can control their schedule completely, maintaining consistent sleep and wake times when possible, getting bright light exposure early in the day, and minimizing blue light exposure in the evening can help maintain circadian alignment. Cardiovascular health influences glymphatic function through its effects on arterial pulsations. Remember that the pumping action created by heartbeats helps drive cerebrospinal fluid through the brain. Maintaining cardiovascular fitness through regular exercise supports robust arterial pulsations. Studies have shown that better cardiovascular fitness correlates with markers of better glymphatic function. Exercise may also benefit sleep quality more generally, making it easier to fall asleep and potentially increasing slow-wave sleep. The type, intensity, and timing of exercise all matter. Moderate aerobic exercise appears particularly beneficial. Exercising too close to bedtime may interfere with sleep for some people, while others tolerate evening exercise well. Finding an exercise routine that fits your schedule and preferences, sustained consistently over time, supports both cardiovascular health and sleep quality. Alcohol consumption presents a complex relationship with sleep. While alcohol may help people fall asleep faster, it significantly disrupts sleep architecture, particularly suppressing REM sleep and causing fragmented sleep in the second half of the night. These disruptions likely impair glymphatic clearance despite the increased time spent asleep. Limiting alcohol, especially avoiding it in the hours before bedtime, supports better sleep quality and more effective brain cleaning. Caffeine, helpful for maintaining alertness during the day, can significantly impair sleep if consumed too late. Caffeine has a half-life of approximately 5 to 6 hours, meaning that caffeine consumed in late afternoon can still substantially affect sleep many hours later. Individual sensitivity to caffeine varies, with some people able to consume it later than others without sleep disruption. However, many people underestimate caffeine's effects, attributing sleep problems to other causes while overlooking their coffee or tea consumption patterns. Limiting caffeine to morning hours, or eliminating it entirely if sleep problems persist, represents a simple change that can substantially improve sleep quality for many people. Body weight affects sleep quality, particularly regarding sleep apnea risk. Excess weight, especially around the neck and upper body, increases the likelihood of airway collapse during sleep, causing or worsening sleep apnea. Weight loss, even modest amounts, can significantly improve or resolve sleep apnea in overweight individuals, leading to better sleep quality and improved glymphatic function. The relationship creates another vicious cycle. Poor sleep impairs metabolism and increases appetite, making weight loss more difficult, while excess weight worsens sleep quality. Breaking this cycle requires addressing both components simultaneously. Medications can significantly affect sleep architecture and quality. Many common medications, including certain antidepressants, blood pressure medications, corticosteroids, and stimulants, can disrupt sleep. If you're taking medications and experiencing sleep problems, discussing this with your healthcare provider may reveal opportunities to adjust timing, dosing, or switch to alternatives with fewer sleep effects. Never discontinue or adjust medications without professional guidance, but recognizing medication effects on sleep as a legitimate concern deserving attention can lead to important improvements. Cognitive behavioral therapy for insomnia, or CBTI, represents an evidence-based treatment for chronic sleep problems that doesn't rely on medications. CBTI addresses the thoughts, behaviors, and environmental factors that perpetuate insomnia. Components typically include sleep restriction, stimulus control, cognitive restructuring, and sleep hygiene education. Research shows CBTI can be highly effective for chronic insomnia, often producing lasting improvements where medications provide only temporary relief. For people with persistent sleep problems that don't resolve with lifestyle modifications, CBTI delivered by a trained therapist represents an important treatment option. Temperature affects sleep quality, with cooler environments generally promoting better sleep. Core body temperature naturally drops during sleep, and facilitating this temperature decline helps initiate and maintain sleep. Keeping your bedroom cool, roughly between 60 and 67 degrees Fahrenheit for most people, supports this natural process. Taking a warm bath or shower before bed paradoxically helps by causing a compensatory drop in core temperature after you emerge. Individual preferences vary, but most people sleep better in cooler rather than warmer environments. Light exposure patterns profoundly influence circadian rhythms and sleep quality. Bright light exposure, especially blue wavelength light, suppresses melatonin production and signals to your biological clock that it's daytime. Getting bright light exposure early in the day helps anchor your circadian rhythm, promoting alertness during the day and better sleep at night. Conversely, limiting bright light exposure, particularly blue light from screens, in the evening allows melatonin levels to rise naturally, facilitating the transition to sleep. While completely avoiding screens before bed may not be practical for everyone, dimming screens, using blue light filters, and keeping evening lighting relatively dim can all support better sleep. Stress management represents another crucial factor for sleep quality. Chronic stress activates the hypothalamic pituitary adrenal axis, leading to elevated cortisol levels that can interfere with sleep initiation and maintenance. Stress also activates the sympathetic nervous system, the body's fight-or-flight response, which opposes the parasympathetic activation necessary for sleep. Developing effective stress management techniques, whether through meditation, mindfulness practices, progressive muscle relaxation, exercise, therapy, or other approaches, can significantly improve sleep quality. The relationship between stress and sleep is bidirectional. Stress disrupts sleep, while poor sleep impairs stress resilience, creating another cycle that requires attention to both components. The concept of sleep pressure offers insight into optimizing sleep timing. Throughout the day, a neurochemical called adenosine accumulates in your brain, creating increasing pressure to sleep. During sleep, adenosine levels decline. This sleep pressure, combined with your circadian rhythm, determines when you feel sleepy and alert. Caffeine works by blocking adenosine receptors, temporarily masking sleep pressure. Napping during the day reduces sleep pressure, potentially making nighttime sleep more difficult. Understanding these dynamics can help you work with your body's natural sleep-wake regulation rather than against it. For most people, avoiding naps or limiting them to brief early afternoon naps helps maintain adequate sleep pressure for good nighttime sleep. Physical health conditions that affect brain function also deserve attention in the context of glymphatic health. Diabetes, hypertension, high cholesterol, and other cardiovascular risk factors not only increase stroke risk, but may also impair glymphatic clearance through effects on cerebral blood flow and vascular health. Managing these conditions through appropriate medical treatment supports brain health both directly and through maintaining glymphatic function. The interconnectedness of body systems means that whole body health supports brain health, and sleep's role in maintaining both highlights its central importance in overall well-being. Hydration status might influence glymphatic function, given the system's dependence on fluid flow. While research specifically examining hydration's effects on glymphatic clearance remains limited, maintaining adequate hydration throughout the day supports general brain function and may facilitate optimal cerebrospinal fluid dynamics. However, drinking excessive fluids immediately before bed can disrupt sleep through nighttime bathroom trips, suggesting that consistent hydration throughout the day rather than evening fluid loading is optimal. Dietary patterns may affect sleep quality and potentially glymphatic function through multiple mechanisms. Certain nutrients support sleep, including magnesium, B vitamins, and tryptophan, an amino acid that's a precursor to serotonin and melatonin. Diets rich in fruits, vegetables, whole grains, and omega-3 fatty acids associate with better sleep quality in epidemiological studies. Conversely, diets high in processed foods, saturated fats, and sugar may impair sleep quality. The timing of eating also matters, with late-night eating potentially disrupting sleep through effects on circadian rhythms and through discomfort from digestion. Establishing regular meal patterns and avoiding heavy meals close to bedtime supports better sleep. For older adults who naturally experience declining sleep quality and reduced glymphatic efficiency, optimizing sleep becomes even more critical. While some age-related changes in sleep architecture may be inevitable, many older adults experience unnecessarily poor sleep due to correctable factors. Sleep disorders like sleep apnea increase in prevalence with age, but often go undiagnosed and untreated. Medications that disrupt sleep become more common with age. Physical health conditions that interfere with sleep become more prevalent. Addressing these factors aggressively in older adults could potentially slow cognitive decline and reduce dementia risk by maintaining better glymphatic clearance despite aging-related impairments. The bedroom environment deserves thoughtful optimization. Beyond temperature and darkness, factors like mattress and pillow quality, noise levels, and electromagnetic fields can all influence sleep quality. Investing in a comfortable, supportive mattress appropriate for your body and preferred sleep position pays dividends in sleep quality. Addressing noise through soundproofing, white noise machines, or earplugs can help for light sleepers. Some people find that minimizing electronic devices in the bedroom, beyond their effects on light exposure, improves sleep through reduced electromagnetic field exposure or simply through psychological effects of creating a technology-free sleep sanctuary. Sleep tracking technology, increasingly accessible through smartphones and wearable devices, offers opportunities to gain insight into your sleep patterns. While consumer sleep trackers aren't as accurate as clinical polysomnography, they can provide useful information about sleep duration, consistency, and rough estimates of sleep stages. This data can help you identify patterns, evaluate the effects of changes you make, and recognize when professional evaluation might be warranted. However, excessive focus on sleep metrics can sometimes create anxiety that interferes with sleep itself, a phenomenon sometimes called orthosomnia. Using sleep tracking as one tool among many, rather than becoming obsessed with optimizing every metric, represents a balanced approach. Professional evaluation becomes important when sleep problems persist despite reasonable self-help measures. Sleep specialists can diagnose conditions like sleep apnea, restless leg syndrome, periodic limb movement disorder, and other sleep disorders that require specific treatments. Polysomnography, an overnight sleep study, provides comprehensive information about sleep stages, breathing, movements, and other physiological parameters. For people with chronic insomnia, cognitive behavioral therapy for insomnia delivered by a trained provider offers evidence-based treatment. Recognizing sleep problems as deserving professional attention rather than as something to simply endure represents an important perspective shift. The societal changes needed to support healthy sleep extend beyond individual actions. School start times that conflict with adolescent circadian rhythms, work schedules that demand long hours or shift work, cultural norms that glorify sleep deprivation as a sign of productivity, and economic pressures that make adequate sleep a luxury many cannot afford all create barriers to healthy sleep at population levels. Advocating for policies that support sleep, educating others about sleep's importance, and challenging the cultural devaluation of sleep all contribute to creating environments where healthy sleep is possible and valued. Ultimately, optimizing glymphatic clearance requires recognizing sleep as a biological necessity rather than as optional or as something to minimize. The brain's nightly cleaning cycle isn't a luxury, but a fundamental requirement for maintaining cognitive function and preventing neurodegeneration. Every choice that affects sleep quality or duration has consequences for brain health, some immediate and some accumulating over years or decades. Treating sleep as the vital process it is, prioritizing it appropriately, and taking concrete steps to optimize it represents some of the most important investments you can make in your long-term cognitive health and overall well-being. Given the critical importance of glymphatic clearance for brain health, the question naturally arises: what can you do to optimize this process? While research in this area continues to evolve, several factors have been identified as influencing glymphatic efficiency, offering practical strategies for supporting your brain's nightly cleaning cycle. Sleep position represents one of the more surprising findings about glymphatic function. Research in rodents found that sleeping in a lateral position, lying on the side rather than on the back or stomach, enhanced glymphatic clearance compared to other positions. The mechanism likely involves effects on the flow dynamics of cerebrospinal fluid, with the lateral position apparently providing more efficient drainage pathways. In humans, most people naturally shift positions throughout the night, but those who can comfortably sleep on their side may be optimizing their glymphatic clearance. Interestingly, surveys of sleep positions across cultures find that side sleeping is most common, suggesting that humans may have naturally gravitated toward positions that support this important biological function. Sleep duration stands as perhaps the most straightforward factor affecting glymphatic clearance. The system needs adequate time to clear the waste accumulated during waking hours. While individual needs vary somewhat, most adults require 7 to 9 hours of sleep per night for optimal health. Consistently sleeping less than this allows incomplete waste clearance, with each night's deficit potentially accumulating over time. Unfortunately, modern lifestyles often normalize insufficient sleep, with many people sleeping well below recommended amounts. Prioritizing sleep duration, treating it as non-negotiable rather than as something to sacrifice when other demands arise, represents a fundamental investment in brain health. Sleep quality matters as much as quantity. Fragmented sleep, interrupted by frequent awakenings or respiratory disruptions, impairs glymphatic function even if total time in bed seems adequate. Factors that fragment sleep include sleep disorders like sleep apnea and restless leg syndrome, environmental disruptions like noise or light, excessive alcohol consumption, certain medications, and anxiety or stress. Addressing these factors can substantially improve sleep quality. For sleep disorders, professional evaluation and treatment are essential. For environmental factors, creating an optimal sleep environment can help. A quiet, dark, cool bedroom promotes uninterrupted sleep. For stress and anxiety, developing relaxation techniques and addressing underlying causes can improve sleep continuity. The timing of sleep relative to your circadian rhythm influences sleep quality and glymphatic efficiency. Your biological clock, synchronized primarily by light exposure, determines when your body is primed for sleep. Sleeping at consistent times that align with your natural circadian tendencies supports better sleep architecture and more efficient restorative processes. Irregular sleep schedules, frequent time zone changes, and shift work that requires sleeping during biological day all disrupt circadian alignment and may impair glymphatic function. While not everyone can control their schedule completely, maintaining consistent sleep and wake times when possible, getting bright light exposure early in the day, and minimizing blue light exposure in the evening can help maintain circadian alignment. Cardiovascular health influences glymphatic function through its effects on arterial pulsations. Remember that the pumping action created by heartbeats helps drive cerebrospinal fluid through the brain. Maintaining cardiovascular fitness through regular exercise supports robust arterial pulsations. Studies have shown that better cardiovascular fitness correlates with markers of better lymphatic function. Exercise may also benefit sleep quality more generally, making it easier to fall asleep and potentially increasing slow-wave sleep. The type, intensity, and timing of exercise all matter. Moderate aerobic exercise appears particularly beneficial. Exercising too close to bedtime may interfere with sleep for some people, while others tolerate evening exercise well. Finding an exercise routine that fits your schedule and preferences, sustained consistently over time, supports both cardiovascular health and sleep quality. Alcohol consumption presents a complex relationship with sleep. While alcohol may help people fall asleep faster, it significantly disrupts sleep architecture, particularly suppressing REM sleep and causing fragmented sleep in the second half of the night. These disruptions likely impair lymphatic clearance despite the increased time spent asleep. Limiting alcohol, especially avoiding it in the hours before bedtime, supports better sleep quality and more effective brain cleaning. Caffeine, helpful for maintaining alertness during the day, can significantly impair sleep if consumed too late. Caffeine has a half-life of approximately 5 to 6 hours, meaning that caffeine consumed in late afternoon can still substantially affect sleep many hours later. Individual sensitivity to caffeine varies, with some people able to consume it later than others without sleep disruption. However, many people underestimate caffeine's effects, attributing sleep problems to other causes while overlooking their coffee or tea consumption patterns. Limiting caffeine to morning hours, or eliminating it entirely if sleep problems persist, represents a simple change that can substantially improve sleep quality for many people. Body weight affects sleep quality, particularly regarding sleep apnea risk. Excess weight, especially around the neck and upper body, increases the likelihood of airway collapse during sleep, causing or worsening sleep apnea. Weight loss, even modest amounts, can significantly improve or resolve sleep apnea in overweight individuals, leading to better sleep quality and improved lymphatic function. The relationship creates another vicious cycle. Poor sleep impairs metabolism and increases appetite, making weight loss more difficult, while excess weight worsens sleep quality. Breaking this cycle requires addressing both components simultaneously. Medications can significantly affect sleep architecture and quality. Many common medications, including certain antidepressants, blood pressure medications, corticosteroids, and stimulants, can disrupt sleep. If you're taking medications and experiencing sleep problems, discussing this with your healthcare provider may reveal opportunities to adjust timing, dosing, or switch to alternatives with fewer sleep effects. Never discontinue or adjust medications without professional guidance, but recognizing medication effects on sleep as a legitimate concern deserving attention can lead to important improvements. Cognitive behavioral therapy for insomnia, or CBTI, represents an evidence-based treatment for chronic sleep problems that doesn't rely on medications. CBTI addresses the thoughts, behaviors, and environmental factors that perpetuate insomnia. Components typically include sleep restriction, stimulus control, cognitive restructuring, and sleep hygiene education. Research shows CBTI can be highly effective for chronic insomnia, often producing lasting improvements where medications provide only temporary relief. For people with persistent sleep problems that don't resolve with lifestyle modifications, CBTI delivered by a trained therapist represents an important treatment option. Temperature affects sleep quality, with cooler environments generally promoting better sleep. Core body temperature naturally drops during sleep, and facilitating this temperature decline helps initiate and maintain sleep. Keeping your bedroom cool, roughly between 60 and 67 degrees Fahrenheit for most people, supports this natural process. Taking a warm bath or shower before bed paradoxically helps by causing a compensatory drop in core temperature after you emerge. Individual preferences vary, but most people sleep better in cooler rather than warmer environments. Light exposure patterns profoundly influence circadian rhythms and sleep quality. Bright light exposure, especially blue wavelength light, suppresses melatonin production and signals to your biological clock that it's daytime. Getting bright light exposure early in the day helps anchor your circadian rhythm, promoting alertness during the day and better sleep at night. Conversely, limiting bright light exposure, particularly blue light from screens, in the evening allows melatonin levels to rise naturally, facilitating the transition to sleep. While completely avoiding screens before bed may not be practical for everyone, dimming screens, using blue light filters, and keeping evening lighting relatively dim can all support better sleep. Stress management represents another crucial factor for sleep quality. Chronic stress activates the hypothalamic pituitary adrenal axis, leading to elevated cortisol levels that can interfere with sleep initiation and maintenance. Stress also activates the sympathetic nervous system, the body's fight-or-flight response, which opposes the parasympathetic activation necessary for sleep. Developing effective stress management techniques, whether through meditation, mindfulness practices, progressive muscle relaxation, exercise, therapy, or other approaches, can significantly improve sleep quality. The relationship between stress and sleep is bidirectional. Stress disrupts sleep, while poor sleep impairs stress resilience, creating another cycle that requires attention to both components. The concept of sleep pressure offers insight into optimizing sleep timing. Throughout the day, a neurochemical called adenosine accumulates in your brain, creating increasing pressure to sleep. During sleep, adenosine levels decline. This sleep pressure, combined with your circadian rhythm, determines when you feel sleepy and alert. Caffeine works by blocking adenosine receptors, temporarily masking sleep pressure. Napping during the day reduces sleep pressure, potentially making nighttime sleep more difficult. Understanding these dynamics can help you work with your body's natural sleep-wake regulation rather than against it. For most people, avoiding naps or limiting them to brief early afternoon naps helps maintain adequate sleep pressure for good nighttime sleep. Physical health conditions that affect brain function also deserve attention in the context of glymphatic health. Diabetes, hypertension, high cholesterol, and other cardiovascular risk factors not only increase stroke risk, but may also impair glymphatic clearance through effects on cerebral blood flow and vascular health. Managing these conditions through appropriate medical treatment supports brain health both directly and through maintaining glymphatic function. The interconnectedness of body systems means that whole body health supports brain health, and sleep's role in maintaining both highlights its central importance in overall well-being. Hydration status might influence glymphatic function, given the system's dependence on fluid flow. While research specifically examining hydration's effects on glymphatic clearance remains limited, maintaining adequate hydration throughout the day supports general brain function and may facilitate optimal cerebrospinal fluid dynamics. However, drinking excessive fluids immediately before bed can disrupt sleep through nighttime bathroom trips, suggesting that consistent hydration throughout the day rather than evening fluid loading is optimal. Dietary patterns may affect sleep quality and potentially glymphatic function through multiple mechanisms. Certain nutrients support sleep, including magnesium, B vitamins, and tryptophan, an amino acid that's a precursor to serotonin and melatonin. Diets rich in fruits, vegetables, whole grains, and omega-3 fatty acids associate with better sleep quality in epidemiological studies. Conversely, diets high in processed foods, saturated fats, and sugar may impair sleep quality. The timing of eating also matters, with late-night eating potentially disrupting sleep through effects on circadian rhythms and through discomfort from digestion. Establishing regular meal patterns and avoiding heavy meals close to bedtime supports better sleep. For older adults, who naturally experience declining sleep quality and reduced glymphatic efficiency, optimizing sleep becomes even more critical. While some age-related changes in sleep architecture may be inevitable, many older adults experience unnecessarily poor sleep due to correctable factors. Sleep disorders like sleep apnea increase in prevalence with age, but often go undiagnosed and untreated. Medications that disrupt sleep become more common with age. Physical health conditions that interfere with sleep become more prevalent. Addressing these factors aggressively in older adults could potentially slow cognitive decline and reduce dementia risk by maintaining better glymphatic clearance despite aging-related impairments. The bedroom environment deserves thoughtful optimization. Beyond temperature and darkness, factors like mattress and pillow quality, noise levels, and electromagnetic fields can all influence sleep quality. Investing in a comfortable, supportive mattress appropriate for your body and preferred sleep position pays dividends in sleep quality. Addressing noise through soundproofing, white noise machines, or earplugs can help for light sleepers. Some people find that minimizing electronic devices in the bedroom, beyond their effects on light exposure, improves sleep through reduced electromagnetic field exposure or simply through psychological effects of creating a technology-free sleep sanctuary. Sleep tracking technology, increasingly accessible through smartphones and wearable devices, offers opportunities to gain insight into your sleep patterns. While consumer sleep trackers aren't as accurate as clinical polysomnography, they can provide useful information about sleep duration, consistency, and rough estimates of sleep stages. This data can help you identify patterns, evaluate the effects of changes you make, and recognize when professional evaluation might be warranted. However, excessive focus on sleep metrics can sometimes create anxiety that interferes with sleep itself, a phenomenon sometimes called orthosomnia. Using sleep tracking as one tool among many, rather than becoming obsessed with optimizing every metric, represents a balanced approach. Professional evaluation becomes important when sleep problems persist despite reasonable self-help measures. Sleep specialists can diagnose conditions like sleep apnea, restless leg syndrome, periodic limb movement disorder, and other sleep disorders that require specific treatments. Polysomnography, an overnight sleep study, provides comprehensive information about sleep stages, breathing, movements, and other physiological parameters. For people with chronic insomnia, cognitive behavioral therapy for insomnia delivered by a trained provider offers evidence-based treatment. Recognizing sleep problems as deserving professional attention rather than as something to simply endure represents an important perspective shift. The societal changes needed to support healthy sleep extend beyond individual actions. Schools start times that conflict with adolescent circadian rhythms, work schedules that demand long hours or shift work, cultural norms that glorify sleep deprivation as a sign of productivity, and economic pressures that make adequate sleep a luxury many cannot afford all create barriers to healthy sleep at population levels. Advocating for policies that support sleep, educating others about sleep's importance, and challenging the cultural devaluation of sleep all contribute to creating environments where healthy sleep is possible and valued. Ultimately, optimizing glymphatic clearance requires recognizing sleep as a biological necessity rather than as optional or as something to minimize. The brain's nightly cleaning cycle isn't a luxury, but a fundamental requirement for maintaining cognitive function and preventing neurodegeneration. Every choice that affects sleep quality or duration has consequences for brain health, some immediate and some accumulating over years or decades. Treating sleep as the vital process it is, prioritizing it appropriately, and taking concrete steps to optimize it represents some of the most important investments you can make in your long-term cognitive health and overall well-being. As we come to the end of this journey through the sleeping brain, a picture emerges of sleep not as passive downtime, but as an active, essential biological process performing functions that cannot occur during waking. The glymphatic system's nightly cleaning cycle, washing away toxic proteins and metabolic debris, represents just one component of sleep's restorative work. Memory consolidation transforms daily experiences into lasting knowledge. Synaptic homeostasis maintains the delicate balance that allows learning while preventing saturation. Immune enhancement, hormonal regulation, cellular repair, and countless other processes all coordinate during these precious hours of rest. The waste products your brain accumulates during each waking hour, the beta-amyloid plaques and tau tangles that threaten cognitive health if allowed to persist, the metabolic debris and inflammatory molecules and damaged proteins, all require clearance. The glymphatic system, that elegant network of perivascular spaces and astrocyte channels, provides the mechanism for this clearance. But it requires sleep, proper sleep with adequate slow-wave activity and sufficient duration to function effectively. Without this nightly cleaning, the brain gradually accumulates the molecular garbage that eventually impairs function and potentially triggers devastating neurodegenerative disease. The evidence linking sleep to brain health has grown from suggestive to compelling over recent years. We now understand that chronic sleep deprivation doesn't merely cause temporary fatigue, but poses genuine long-term risks to cognitive function and brain integrity. The cognitive impairments, the emotional dysregulation, the increased dementia risk, and the accelerated brain aging associated with poor sleep all reflect real biological consequences of inadequate glymphatic clearance and impaired restorative processes. Conversely, prioritizing sleep represents one of the most powerful interventions available for protecting brain health across the lifespan. The practical implications are clear. Sleep isn't optional. It isn't something to sacrifice casually in pursuit of productivity, entertainment, or social pressures. The hours you spend sleeping aren't wasted but invested, returning dividends in cognitive performance, emotional stability, physical health, and longevity. Optimizing sleep through attention to duration, quality, timing, sleep position, and the numerous other factors we've explored represents concrete action you can take today to support your brain health tomorrow and decades from now. For those who struggle with sleep, whether due to insomnia, sleep disorders, chronic stress, or environmental factors, seeking solutions deserves high priority. The consequences of chronic poor sleep are too significant to ignore or accept as inevitable. Professional evaluation, appropriate treatments, lifestyle modifications, and persistent attention to sleep can yield improvements that cascade beneficially through every aspect of health and functioning. The societal implications extend beyond individual health. A population that chronically sacrifices sleep operates below its cognitive potential, makes poorer decisions, experiences more mental health problems, faces higher risks for chronic disease, and incurs substantial economic costs through healthcare expenses and lost productivity. Creating societies that value and support adequate sleep requires cultural shifts, policy changes, and collective recognition of sleep's importance. From school start times to work schedules to cultural narratives about success and productivity, numerous aspects of modern life could be restructured to better support rather than undermine healthy sleep. The science of sleep continues to advance rapidly, with new discoveries emerging regularly. Our understanding of the glymphatic system, discovered only in 2012, continues to deepen. New imaging techniques allow researchers to observe brain processes during sleep with unprecedented detail. Interventions to enhance slow-wave sleep, to improve glymphatic clearance, or to protect against the consequences of sleep deprivation are being developed and tested. As this science progresses, new opportunities to protect and enhance brain health through sleep optimization will emerge. But we don't need to wait for future discoveries to act on what we already know. The evidence already compels us to treat sleep as the biological necessity it is. Every night you sleep well, you clear toxic proteins from your brain, consolidate important memories, recalibrate your synapses, strengthen your immune system, regulate your metabolism, and restore your capacity to function optimally. Every night represents an opportunity to invest in health across multiple domains simultaneously. As you prepare for sleep tonight and every night, remember that you're not simply resting. You're activating one of the most important maintenance programs your body possesses. Your brain is preparing to expand the spaces between its cells, to flush cerebrospinal fluid through its tissue, to wash away the day's accumulation of molecular waste. Your neurons are getting ready to replay the day's experiences, integrating them into lasting memories while pruning unnecessary details. Your body is preparing to repair DNA damage, to synthesize proteins, to enhance immune function, and to regulate hormones. All of these processes coordinate in the elegant choreography of sleep, processes that cannot be skipped or shortchanged without consequence. The next time you're tempted to sacrifice sleep for one more hour of work, one more episode of a show, or one more scroll through social media, remember what you'd be giving up. Remember the glymphatic system waiting to clean your brain, the memories waiting to be consolidated, the synapses waiting to be recalibrated. Remember the long-term cognitive health at stake, the accumulating risk of every night of inadequate sleep. And then, perhaps, you'll make the choice to prioritize sleep, to give your brain the time it needs to clean itself, to maintain itself, to prepare itself for tomorrow. Your brain, that 3-pound universe of neurons and glial cells, chemicals and electrical signals, has carried you through every experience of your life. It deserves the nightly maintenance that only sleep can provide. The glymphatic system stands ready each night to perform its essential work, but it requires your cooperation in the form of adequate, high-quality sleep. Give it that opportunity. Prioritize sleep. Optimize your sleep environment and habits. Seek help for sleep problems when needed. Treat sleep as the precious, irreplaceable resource it is. The sleeping brain is not an idle brain but a working brain, engaged in maintenance that preserves function and prevents disease. Understanding this transforms how we think about those hours of unconsciousness. They're not lost time, but invested time, not passive rest, but active restoration. They represent a biological necessity as fundamental as food or water, a requirement that cannot be negotiated away or indefinitely deferred. Embrace sleep. Protect it. Optimize it. Your brain, cleaning itself each night while you dream, will thank you for it with decades of optimal function, preserved memories, and protected cognitive health. If you found this information valuable and want to learn more about how sleep affects your brain and health, please consider subscribing to the Sleeping Subconscious. Hit that like button if this video helped you understand the remarkable processes happening in your brain every night. And remember, the next time you lay your head down to sleep, you're not just resting. You're giving your brain the gift of restoration it absolutely needs to thrive. Good night, sleep well, and let your glymphatic system do its essential work.